Hello, everyone. My name is Lu Liu, and I'm assistant professor of North Dakota State University in the Department of Computer Science. And today, I want to give a short introduction to bioinformatics and bioinformatics research. Look at the agenda, and first, I want to talk about what's bioinformatics, then resources used in my lab, and last, briefly about my research. Bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data. Look at the figure. It's a typical bioinformatics workflow. Researchers and scientists devise some experiments, and the experiment materials feed into the next generation sequencing machines, and then bioinformatics pipelines used to analyze the data generated by the machine, and the pipelines also will generate some kind of the visualization result. For example, for here, there's a heat map, and the heat map can be used for knowledge discovery and publication. At the same time, they can feed back to the experiment design to evaluate the experiment. You can see that bioinformatics play an important role in the middle. And in bioinformatics, we are very interested in the genome. And a genome is an organism's complete set of the DNA. I think you already see picture like this in movies, and they represent a genome. And for human genome, there are 23 pairs of chromosomes from chromosome 1 to chromosome 22. And if you are female, and then you have two X's. If you are male, you have X and Y. And for our human genome, and we have 3 billions of the basic units. We call them bases. So it's a very, very large number. And for the first human genome project, the goal is to determine the sequence of these 3 billion bases. The figure just shows you a very small portion of the sequence, and it took about 13 years and cost about $2.7 billion. With the development of technology, there are large machines like this, and they can run in a very short time and in a single run to get a lot of data and save us a lot of the time. And also the cost of the sequencing a genome decreased dramatically from the very beginning, and it cost about 100 million to sequence one genome until to very recently, and the cost is decreased to close to $1,000. And for the bioinformatics, and there is a lot of the applications. And for example, and we can use it in disease diagnosis, and we can use some kind of the bioinformatics to scan patients and to help us to identify the patients have a certain type of disease or not. And also in future, we can use it for personalized medicine. In future, doctor prescribe medicine or treatment according to our DNA information. And the third application is in forensics, and policemen can use the DNA information to identify criminals, and also can be applied in agriculture. And we can use bioinformatics to help us to acquire plants with desired properties, such as high yield, enriched with some kind of the nutritions, or pest resistance. So there's a lot of the applications. And with the development of the application and also the tool, and bioinformatics generating a lot of the data, and you can see that. And at the very beginning, the number of the sequence is 0, and you can see that it increased tremendously or exponentially. And now we move into a big data era, and we need to have a very powerful computers for us to process the data. And then I want to talk about some of the resources used in my lab. And because we are moving into big data era, and there's a lot of the biological data, and we need to have powerful computers for us to do it. And at NDSU, we have a very strong cluster. It's called the Center for Computational Assisted Science and Technology. We call it CCAST. And in my lab, we use this cluster to help us to analyze computing intensive tasks. And also for this cluster has very large memory and large storage. In our lab, and we using some open source software, and the first one is a Python, and it's very powerful and also very popular. And if you are interested in programming, and I would recommend you to learn Python first. And along with the Python, there are another software is called Anaconda, and this one contains a lot of the open source free Python package, and it's provide a lot of the functions, and also some of the tools help you to write, debug, and execute your Python program. And we also use GitHub, and it's very popular to storage your code and share with your teammates, and also share with the other people. And we are doing research in bioinformatics. We need data, and generally, and we get data from two sources, and we either get data from our collaborators. They will do the experiments and give data to us. Also, we can download data from public databases. Here are two famous databases. The first one is called ENCODE. The second one is called SRA, and both of them accumulate a lot of the DNA data. And if you're interested, you can Google them and you can look at them. And now I want to mention about my research. My first research is differential analysis of chromatin interactions. And I want to explain the terminology here. And for differential analysis, and which is a very common task, and it's try to compare two groups of subjects and to identify the differences. And it's similar to the figure, and you can see that there are two figures, and we try to identify the difference between these two figures. And it's very similar to that, and but we are not working on pictures. We are working on chromatin. And the chromatin, and it's a basic structure unit of chromosome, and it's a complex of DNA and protein. In the figure, you can see that there's a cell. Inside of cell, there's a nucleus. In the nucleus, there are chromatin, and they look like spaghettis, and they are packed in a very complex and dense way. And in chromatin, there's a phenomenon. It's called the chromatin interaction. And for the chromatin interaction, it means, and there are some locations on the chromosome from one chromosome or two different chromatin. They can bind to each other through some molecules. And in our research, we try to identify the difference on chromatin interactions between two groups. And you can see that, and our method, and actually is try to taking data in, and then try to process them step by step, and finally to report some of the locations, and they are significantly different between two groups. And in the middle, actually, it's boiled down to compare two networks. And for example, there are two networks, and they share the same nodes, but the connections between the nodes are different. For example, if you look at the node D in these two networks, and you'll find out the node D connect to node A and node C, and the connection in these two networks are the same. But if you look at the node B, and you'll find out in network 1, and the B is connected to node A, but in the second network, it's connected to node C. So the connection is significantly changed because they are connecting to different nodes in two networks. And in a simple network, it's easy to examine manually or by eyes, but in a large network, it's impossible to do that. And we need to have some kind of the mathematical way to find out, to help us to identify nodes. And we will resort to some kind of measurements and help us to find out, for example, for the node B, and you will get a larger value. For node D, you will get a smaller value. Large value means this node is significant different between two groups. For small value, it means they are almost the same in two groups. And another type of research, and I try to identify the relationship between gene sets. And on the left, you will see some genes, and these genes is called input genes. They are generated from some experiments, and the biologists want to understand them because they may have some kind of function they haven't known yet. For the gene on the right, represent a pathway. A pathway refers to some knowledge, and it represents some kind of function. For example, maybe they represented the DNA replication function, something like that. So we want to establish some relationship from the input genes to pathways. And to do that, and we try to resort to networks. And in the middle, there's a network, and each node represents one gene. And at the first step, and we try to map the input genes to the network. At the same time, try to map the genes in the pathways, different kind of pathways to the network. And then we try to propagate some of the values at the input genes to other genes through a mathematical formula. It's called the random walk with restart. And then for each node in the network, will receive some values. And then we can calculate the values in different pathways, and then we can do some kind of way to measure the relationship from the input genes to pathways. And for this research, we have an application. We collaborate with some cancer scientists on endometrial cancer, and we find out 5 new pathways, and they are statistically significant, and we publish this to as a paper in a very good place. And in my other research, we also try to build databases and bioinformatics tools to help other people. And for example, and the first picture is a database for Arabidopsis. It's a plant, and we try to identify the class and module and the pattern in Arabidopsis. The second one contains 2 tools, and they are based on the chromatin interaction. And if you're interested, and there are 2 links at the bottom, and you can access them and play with them. And I think that's what's about bioinformatics and what kind of research I'm doing, and also what kind of tools I use. If you're interested, you can send an email, and I'm very happy to receive an email. And thank you.